mention, might not be a big deal, but over the break I realized that I had some things organized wrong in the Blackboard stuff, and so I fixed that. Last summer, I had the blood chapter stuff in this folder because of how I arranged the semester last year. So those are no longer in there, along with the autonomic material in Unit 2, and part 1, 2, 3, and lab quiz for that. There's also the endocrine system parts 1, 2, 3, and 4, and its lab quiz. So we are doing the 2 units this week together, so all of those assignments are going to be due. And I just realized I probably ought to double check the due dates in the system for those 2. But, if you look at the due date right now, it might not say it, but the autonomic system and the endocrine system due dates, as soon as I get a chance to go in and double check them, we'll say that they're all due by midnight next Tuesday. So you need to do that. So what I want to concentrate on today, sort of like using the case study in the first half, I want to show endocrine system material in micrographs. And I have a link in this unit's folder that says endocrine system micrographs. These are pictures that I took with this scope up front of the slides we have in our slide set here. I used to have students get their microscopes out, and I'd show the same stuff on the screen as I was talking about it, and then eventually I just said, well, why don't I just start capturing these pictures and have it here, so you don't have to get the microscope set out. If you're interested in looking at these slides yourself in more depth, you're welcome to do that. We'll have the microscopes out for some stuff. Let's see. Actually, no, I guess we won't get them out in the next couple of weeks. That'll be later on in the semester. If you want to look at the microscope slides for this stuff on your own, the best way to do that is room 511 down the hall is the A&P study room. It has most everything that's in the lab down there, all the models and stuff that we have. And we also have a full set of the slides down there. We keep that room locked, but because we don't want people walking away from any of that stuff. But we'll open it for any of the A&P students in the department. It's not used for teaching. It's only available for you to study from. So you're welcome to go in there if you want to. So after class or something like that, if you want to spend some time studying, I'll unlock the door for you. If you come in early in the morning, if you want to spend a little time in there before class, myself or one of my colleagues can open the door for you. Professor Machazik, who teaches micro, he comes in like butt early every day. So he's probably here like at 6 o'clock or something like that. And you're welcome to pastor him. And if you want to come in, some other day of the week that I'm not here, one of my colleagues can help. If you can't find one of the professors on this floor, upstairs in room, I think it's 603, which is the prep room, one of the lab techs can open the door for you. That's what the room is there for you to study. The slides I'm going to show you today of the endocrine system are all out of the yellow slide box. And I've labeled the pictures in this folder so you know what we're looking at. Also, I'm going to be showing you multiple magnifications. So in each image, the title for each image, it tells you which objective magnification it's based on. I don't have the total magnification because I'm actually not positive about it. If we're looking through one of these scopes, total magnification is very easy to figure out because the oculars are 10x and then the objectives are 4, 10, or 40x. And so you just multiply the two together. It's just the camera. I'm not positive about the magnification factor on the camera, so I don't know what the total magnification of this is. So I'm just giving you the objective magnification, just because it's a relative idea here. Just, is it a high power, low power, et cetera, kind of thing. In the yellow box, these are slides 11, 12, 13, and 14. I'm going to go through them in that order. So if you want to look at them on your own at some point, just whichever of the slides we're talking about here, it'll be in that order there. In talking about this, I want to talk about some of the concepts for the endocrine system that are important. Before I actually start talking about these particular glands, I just want to talk about what the endocrine system is and what those glands are. Let me use this torso model for a second, just to point this out. This isn't something I can capture on, oh, actually I could do it this way. I'll use the model there in a second, but actually, come to think of it, you just go to the endocrine system part of the book, and this is just a picture that kind of depicts the same thing. So the major endocrine glands that we tend to think of when we're considering the system, I'm going to sort of work through them from the top down. In the cranial space, there are three structures that we're interested in considering. One is the pineal gland. Pineal is referring to the fact that it's shaped kind of like a pine cone. That's where the name comes from. It's posterior to the thalamus. It actually is also sometimes referred to as the epithalamus. All it does is it releases a hormone called melatonin, which is the chemical signal in your body for what time of day it is. Now, it's not like a clock. It can't tell you that it's 11 or 12 or something like that. It's rather just the difference between day and night. So at night, when the sun has gone down, the pineal gland releases melatonin. And then in the morning, when the sun comes up, it stops releasing melatonin. So your body knows whether it's day or night based on that. It also helps lock us into the 24-hour cycle of the sun and moon, that sort of thing. It's responsible for controlling the circadian or diurnal rhythms of your body. Yeah.
Does it switch if you were to work like third shift?
The way it works is your visual system is receiving information about the light that you're seeing.
Okay.
And when sunlight, which is broad spectrum white light, hits your retina, that's processed through the hypothalamus and it tells the pineal gland it's day. And then when light levels dim for night, then and you stop getting that broad spectrum sunlight, then your pineal gland knows it's dark. And
So what about if you lived in Alaska? Yeah, I was just joking.
Then you get really screwed. Yeah. Seasonal and affective disorder is when the seasons change the daylight cycle enough. So here, even at this latitude, when it's the difference between summer when the sun's going down almost at 9 o'clock versus winter when it's going down at 4:30, people will have more melatonin in their system in the winter, and that'll lead to mood disorders. So if you're at a very Arctic or Antarctic elevation, latitude, then it's going to be even thrown off even more. So if you're in the eternal sunlight, then you're never going to have a melatonin release, and it might throw off sleeping schedules and that sort of thing. Or if it's winter and the sun never comes up, then you're just going to have sort of this constant melatonin levels in your body. And so the pineal gland does sometimes have something to do with mood disorders. One way to fix that is to just fake it out. You can buy a light that's broad spectrum white light and make your body think that the sun's out even if it's not. So that's a kind of quick fix to some of the more moderate seasonal affective disorder kind of things.
Really? The white light will help you?
Yeah. While it's not actually pointed out in this picture, it's actually crucial to what's happening. Right here, so in this picture, they point to the pineal gland. and the pituitary gland, which we'll talk about in a second. And then they have a bracket showing where the thalamus is located. It's sort of like they're showing everything but the most important thing, which is the hypothalamus. So where the pineal gland's attached here, that's essentially where the hypothalamus is. And the hypothalamus is very important for the endocrine system. Actually, it's very important for the endocrine and autonomic systems. Autonomic function is regulated by the hypothalamus, as is endocrine function. The hypothalamus is really in charge of homeostatic mechanisms, and it uses autonomic or endocrine function to regulate that. And then we have the pituitary gland, which is the primary way that the hypothalamus controls the endocrine aspect of homeostatic mechanisms. Sorry, I was about to say autonomic, which would be confusing. The hypothalamus will release hormones that tell the anterior pituitary to release hormones, which will tell other glands in your body to do their jobs. So it's sort of this master control system through that. Then as we move out of the cranial space, next we get to the thyroid, which is located right at the base of the neck, right here, wraps around the esophagus. It's sort of at the level of the larynx. When we learn about the larynx, the cartilage wall of the larynx is called the thyroid cartilage because it's anatomically right next to that, which is depicted here. If we were to rotate the thyroid gland around and look at its posterior surface, we would see the parathyroid glands. Now in this picture, the thyroid gland, and this is a posterior view of it, looks like it's connected in the back. In fact, that wouldn't necessarily be true. There is some variation from person to person, so there might be some people that have a thyroid gland that wraps all the way around the trachea, but more often it doesn't quite meet on the posterior side. But on the posterior side of the thyroid gland are these small parathyroid glands, which for the sake of a picture like this, they're exaggerated. They're not nearly that large. They're very, very small little flecks of tissue. We're going to look at that in the microscope in a little while. And then moving down from that, we get to the thymus gland, which is sitting, again, kind of anterior to the esophagus, or trachea and esophagus, as well as the heart. Now, if we look at a model like this torso model here, we don't see the thymus, and they leave it out on purpose. There's two reasons for that. One, it blocks our view of what's sort of the most important thing that anatomists usually want to look at, which is the heart. So it would be sitting on top of it, blocking our view of the heart. Also, this is a model of an adult human. And in the adult, the thymus gland is no longer really active. It's only active during childhood and development, childhood and adolescence. It's important for the development of the immune system. So once the immune system is fully developed, then it really doesn't have any role to play, and it stops becoming a functional gland. So they just leave it out of a torso model like I have on the table over there because it would just get in the way of seeing the heart. But that's where it's located. And then we jump down to the adrenal glands, which I mentioned earlier because the adrenal medulla is part of the sympathetic system, sort of. It releases epinephrine into the bloodstream when it's stimulated by preganglionic parasympathetic fibers. The adrenal cortex, the outer part of that, is responsible for releasing a bunch of other hormones in the bloodstream. We'll look at that under the microscope in a little while also. And then right next to that, or right in between the kidneys there, we see the pancreas. The pancreas always gets sort of a big starring role in any endocrine system chapter like this, but that's kind of misleading. The pancreas is actually more a digestive gland than an endocrine gland. About 90% of its function is digestive. It makes digestive enzymes and juices that are secreted into the small intestine. Only about 10% of what it does is endocrine in function, but that endocrine function is fairly well known. People are probably pretty familiar with that, so it gets a lot of press, so to speak. And we'll talk about that, looking at the pancreas on the microscope in a while also. And then we move down, and really the rest of what's here is reproductive stuff. This is, I suppose, a hermaphroditic picture here. It has a uterus and ovaries as well as testes. And that's just for the sake of pointing out all of those glands. So the ovaries and the testes are analogous structures, female and male reproductive systems, secreting their respective hormones, whether it's a hormone in the estrogen group or testosterone. And then there's also progesterone, which is released from the ovaries and the uterus, which has a lot to do with gestational function, and that aspect of the reproductive function. Those are sort of our big players when we think of the endocrine system. And today I'm going to show you micrographs of the pituitary gland, the thyroid and parathyroid glands, the pancreas, and the adrenal glands. We're not going to look at the other ones. The thymus we're going to talk about more when we get to the immune system. I've said all I'm going to say about the pineal gland a little bit ago about bright light and mood disorders. And then the reproductive system will cover the ovaries and the testes and the uterus then. That is what we usually think of as the major players in the endocrine system, but it's not really the entire endocrine system. What's left out are a lot of organs that have endocrine function to them, but we usually think of them more about the other functions that they play. For example, the heart releases hormones that help regulate blood pressure. When blood pressure is too high, the atria of the heart, the first chamber that blood goes into, will stretch out because there's too much blood going to it, or it's going in there too fast. And so the heart releases hormones to help limit blood volume and blood pressure so that the heart doesn't have to work too hard. Another example is the kidneys, which are actually in this picture, though they're not labeled. They also release hormones that play a role in blood pressure. They're sort of the opposite side of what I just talked about. The heart tries to limit blood pressure and blood volume so that the blood doesn't have to work too hard, right? So that the heart doesn't have to work too hard. The kidneys, on the other hand, work oppositely. They keep blood pressure up because blood pressure is important for kidney function. And so the heart and the kidneys kind of play against each other using an endocrine functionality there. Another important endocrine function that's left out in this picture is there are endocrine signals in the digestive system that are very important for regulating how the stomach and the digestive aspects of the pancreas and the liver and the small intestine, how all of those work, and they have hormones associated with them too. So that's kind of a quick overview of what's going on in the system. And like I said, we're going to pay attention looking at micrographs for the pineal gland, the thyroid, parathyroid glands, the pancreas, and the adrenal glands. Those are the micrographs that I have to show you, and specifically in that order. Before I get to that, I also want to point out a couple of other things important from the book. When we say endocrine, like I said earlier, endocrine specifically means secreting within the body. We're talking about hormones, and sort of the defining characteristic of what is a hormone is a chemical, signaling chemical that travels through the bloodstream. It's released from one of these glands, and it has an effect somewhere outside, somewhere else in the body, some other tissue. There are other aspects of chemical signaling that really kind of fall into the endocrine system also, but they're referred to slightly differently. The exact same molecules, the same things that we call hormones, will have, as these two paragraphs that I have on the screen here point out, either autocrine or paracrine effects. So again, those two words have crine in them, meaning secretion. Autocrine means self-secreting. So that's when a gland or tissue that secretes a hormone has receptors for that hormone on it, and that it'll respond to its own hormones. A good example of that is in the stomach. In the glands in the stomach that make stomach acid, there are cells that release a hormone called gastrin. And gastrin increases stomach activity. So when you're eating food and your stomach is very active, it's making stomach acid and other secretions to help digest the food. And it makes gastrin, which will tell those glands to make more stomach acid and other digestive juices to make the stomach digest the food faster. The cells that make gastrin are sensitive to gastrin. So when they make gastrin, they basically tell themselves to make more gastrin. And that's an autocrine function. Similarly, paracrine function can be described by that too. That gastrin that's released from those cells is telling the neighboring cells. It's not getting into the bloodstream. It's just right there in that tissue telling the other cells to be more active, to make more acid, or to make more enzymes or something like that. That's a paracrine function. Gastrin is a hormone. It does get into the bloodstream. It does have an effect further away, but it does have local effects on the cells that secrete it itself, which is autocrine, and on its neighboring cells, which is paracrine. So those are other aspects of chemical signaling, which are really part of endocrine function, still secreting within the body. But they aren't necessarily going through the bloodstream, which is sort of our main definition of what a hormone is, something that travels through the bloodstream. So this gives you a list of different hormones and where they're found and some definitions of what they are. I'm going to talk about that a little bit more in terms of the what the tissues I'm going to show you under the microscope in a second. But an important distinction in the types of hormones that are out there, which all the stuff that I just scrolled through in the book right there, talking about the specific types of hormones, doesn't address. But then the book does address it when it gets here. I think the most important distinction to make about hormones is to separate them into groups that are lipid soluble and water soluble. Okay. Now, hormones always travel through the blood, and the blood is a water environment. So lipid soluble hormones need help dissolving into the plasma and traveling through the blood. But then the hormone wants to interact with its target cell. Which means that the cell has a receptor for that hormone. Lipid-soluble hormones can dissolve through the lipid bilayer of the cell membrane. So they get into the cell, which is what this picture on the screen is showing. And the receptor is inside the cell. Now, your concept of a receptor at this point probably is based mostly on what we talked about in the nervous system, neurotransmitter receptors and that sort of thing. This type of receptor is very different from that type of receptor because it's inside the cell. But when the hormone binds to the receptor, they, as a complex, bind to DNA in the cell and change what's going on in the cell. The best example of that is steroid hormones like testosterone, which is referred to as an anabolic steroid. When testosterone gets into a cell and binds to the testosterone receptor, as a complex, the testosterone receptor and testosterone molecule together bind to DNA and increase protein production in that cell. I just described that as an anabolic steroid. When you hear the term anabolic steroid, what do you associate that with? Has anybody ever heard the term anabolic steroid before? Okay, let's just go with steroid. When you hear steroid, what do you think of? Muscles. Why?
Don't a lot of bodybuilders, they
Yeah, steroids can help build muscle. What is muscle made out of primarily? What are those fibers made out of? Protein. Right. Okay. So an anabolic steroid is going to build proteins and therefore increase muscle mass. Okay. So bodybuilders or athletes will often abuse anabolic steroids because it builds muscle mass. I was expecting somebody to go to the most common association with anabolic steroids, which is performance-enhancing drugs. Okay. Asterisks next to athletes' names in halls of fame, that sort of thing, or because they achieved certain records that they're recognized for, but then later on it was found out that they abused things like anabolic steroids to increase performance. I thought everybody'd heard of those stories. Sound familiar?
Yes.
Okay.
Yes.
That's what we're talking about.
What is anabolic?
Anabolic just means any metabolic process that builds. So building proteins. And I'm really sad that you had to ask that question because that's an AMP1 thing. So, okay. So this picture is depicting a hormone that dissolves through the lipid bilayer of the membrane and gets inside and binds to its receptor. The other type of the other type of hormone is water soluble, which means that it can't pass through the cell membrane. And so it has to bind to its receptor on the surface of the cell. And this is probably what you're used to thinking of when I say receptor. Because neurotransmitters are water-soluble signaling molecules also. The difference between a hormone and a neurotransmitter is simply where we find them. Hormones, like I said a second ago, are found in the bloodstream. Neurotransmitters are found at synapses. Okay, that's the difference. When I was talking about the sympathetic system earlier, and I was talking about epinephrine and norepinephrine, it's easiest for students to hear norepinephrine is a neurotransmitter released from postganglionic sympathetic fibers. And epinephrine is the hormone released from the adrenal medulla. Okay. That's an oversimplification. The adrenal medulla actually releases norepinephrine and epinephrine. Okay. About 20% norepinephrine, 80% epinephrine. So that's enough of a difference for me to just concentrate on the epinephrine side of it. And it makes it easier for students. Epinephrine's a hormone in the bloodstream, norepinephrine's a neurotransmitter at synapses. It's not as simplistic as I make it out to be, but that's a good way to sort of think of it. So whether it's a neurotransmitter or a water-soluble hormone, it'll bind to its receptor on the cell surface. For hormones and some neurotransmitters, what happens inside the cell is what this picture depicts. The receptor on the cell surface belongs to a group of receptors called G protein coupled receptors. Okay. Let me just talk about those for a second. So G protein coupled receptors. If you look these up, you'll see the abbreviation GPCR. There are three parts of these receptors. First, there's the receptor protein, which is the thing in the cell membrane that the signaling molecule binds to. And then there's what's called the G protein, which is attached on the intracellular side of the receptor protein, and it hydrolyzes GTP. Now, you're probably used to hearing hydrolyze ATP more commonly. GTP is just a closely related molecule that has 3 phosphates as well as ATP. And you can hydrolyze it and get some chemical energy out of it. When the G protein hydrolyzes GGP, it moves from the receptor protein to the third part, which is called the effector protein, which is usually an enzyme. Those are the major components of the G protein coupled receptor. Okay. So let me go back to the picture. Where was that? Here. Actually, I'll make the picture bigger. In this picture, the receptor protein, which is embedded in the cell membrane, kind of looks like a pimple on the side of this cell. It's a weird drawing, but that's the receptor. It's in the cell membrane. On the intercellular side of the cell membrane, that's where the G protein is. Okay. When in this case, hormone binds the receptor, the G protein becomes activated and it physically moves to interact with the effector protein. Okay. Now in this picture, the effector protein is specifically labeled by this name, which is what this particular effector protein is. But there are other examples of G protein coupled receptors that have different effector proteins. So in the list I just gave you, this is the general term for that third component. And this picture in the book is just showing you a particular example. What the effector protein does is it's going to make a signal inside the cell, sometimes referred to as the second messenger, or as the book refers to it here, the secondary messenger. The second messenger is the internal signal that lets a cell know that the first messenger outside the cell is there. So when the hormone binds, the end result is a new signal shows up inside the cell. That's going to activate some protein enzymes and cause changes inside the cell, not all of which are necessarily depicted in this picture, but several different things can happen. And that changes what happens in the cell. Okay. We're looking at this in the endocrine system chapter. All water-soluble hormones bind to this type of receptor. Okay. This is not the only place we see this type of receptor. Some neurotransmitters also work with this type of receptor. For neurotransmitters, the second messenger is going to eventually cause ion channels to open, and the membrane potential will change and that sort of thing, which is not depicted in this picture because that's not a hormone thing, that's a neurotransmitter thing. But neurotransmitters can also work through a different type of receptor, which is the type that you met last semester, which is an ion channel that opens when a neurotransmitter binds to it. So when you learn about the neuromuscular junction and acetylcholine, you learn that there's a channel that opens when acetylcholine binds to it. Okay. That's not this type of receptor. That's an ion channel itself. Ion channels opening directly because of binding happens practically instantaneously, okay, within microseconds or nanoseconds or something like that. This process takes a little bit longer. It's in the millisecond range, maybe 100 milliseconds, something like that. But it's a little bit slower. Okay. The difference, however, is that this can have a lot of different effects inside the cell, whereas an ion channel just does one thing. It lets ions in. So there's a trade-off between speed and the range of effects. Hormones always need a bunch of different effects, so they always have this type of receptor system. Neurotransmitters can use both when they need a very, very quick response on the scale of nanoseconds or something like that. Then it's going to be an ion channel. When they have the luxury of a slower response, relatively speaking, it's still in the millisecond range of response, they might use this kind of thing. Depends on the neurotransmitter. But sometimes neurotransmitters need a different type of receptor. But all water-soluble hormones have this type of setup. Now, the effector protein can be different That's why I'm giving you that general term for it, while this picture is giving you a specific name for one of them. But in other cells, the receptor might be different. Oh, I thought they had another picture of that here, but they don't. So there can be other receptors that they interact with. So I want to talk a little bit when it comes up in the micrographs I'm about to show you of where water-soluble versus lipid-soluble hormones are found. Because I think that's a very important distinction because of what the target cells are. And what makes something a target cell is simply that it has a receptor. Okay. And I'll talk about that a little bit with some of that in a bit. So let me go to where I have these micrographs stored. You have a link to this page. I want to start by talking about the pituitary gland. Now, the label for this slide is not pituitary gland. It's hypophysis. That's an alternate name for the pituitary gland. The pituitary gland is attached to the hypothalamus. So if we wanted to use a Greek word to describe to name the pituitary gland, since it's below the hypothalamus, we'd want to use some word that has hypo in it. But hypothalamus is the weirdest construction of a Greek word. So that doesn't work. Instead, it's hypophysis. The physis part of this word is an alternate name for the hypothalamus, sort of. And so hypophysis means below the hypothalamus. Okay, just the Greek word for this. When we talk about the pituitary gland, we talk about the anterior and posterior components. When we talk about the hypophysis, we talk about the adenohypophysis and the neurohypophysis. I'm just trying to arrange the words on the screen. So yes, when we're talking about the pituitary gland, we talk about the anterior and posterior components. When we refer to as the hypophysis, we don't use anterior and posterior. Instead, we use adeno and neuro. Okay. So to explain what these words mean, now let me go back to the picture. Okay. What we're looking at here is a micrograph. Now, a theme for all of the micrographs I'm going to show you today is really about what I like to think of as the staining quality of the tissue. Now, by that, what I mean is really how dark or light it is. So let me take a second just to talk about what we're looking at when we have stained tissue. Now, this is a little oversimplified for how stain actually works, but it gets the point across. The stains we tend to look at are what we call cell stains, and they work because the stain reacts with chemicals in the tissue. The stain is essentially a positively charged pigment molecule, and it's going to react with the tissue on a chemical basis because of what it is. What is a positively charged pigment molecule going to react most strongly with?
Negatively charged?
Yeah, a negatively charged molecule. What's the most negatively charged molecule in any cell of your body? You can get to that answer by thinking about it in a different direction. We're talking about stains. You looked at stains. What's the most darkly staining thing in any cell? The nucleus. What does the nucleus contain? The cell nucleus. And electrons are not found in an atomic nucleus anyways. But what is found in a cell nucleus? Again, cell nucleus, not atomic nucleus.
DNA?
DNA, right? That's the answer. The most negatively charged molecule in any cell of your body is DNA. DNA is made up of repeating units called nucleotides. Each nucleotide contains a phosphate group. Phosphate groups are a phosphorus atom and 4 oxygen atoms. Because of how phosphorus and oxygen interact, there ends up being a negative charge to a phosphate group. Each phosphate group, when it's part of a nucleotide, has a -1 charge to it. How negative a DNA molecule is depends on how many nucleotides there are in it. Now, DNA is, by definition, double-stranded. So there's 2 chains of nucleotides. When we talk about the length of DNA, we describe it as base pairs in length. So that's a nucleotide in each strand where they pair up. So a base pair has a -2 charge to it because there's 2 nucleotides, one in either strand for each base pair. The nucleus contains DNA in the form of chromosomes. There are 46 chromosomes in the nucleus of pretty much all the cells in your body. The biggest of the chromosomes is chromosome number 1. There are 2 of them. You got one from mom and one from dad. Chromosome number 1 is 291 million base pairs long. And you have 2 of those. And then chromosome 2, 3, 4, 5, 6, 7, all the way down the line, just have more and more pieces of DNA. They just get smaller and smaller as we count up in the pairs there. So there's a lot of very negatively charged DNA there. What would the charge be for a 291 million base pair long piece of DNA?
Negative.
Yes, it's negative, but how negative?
Very negative.
Can you be more exact?
Could be 2 million.
No. So each nucleotide has a -1 charge. Each base pair has 2 nucleotides in it. 291 base pairs long. No?
-2.
291 times 2.
Yes.
Yeah.
Each base pair has a -2 charge, so 2 times 291 million negative charge.
Oh, wow.
And that's just one of the 46 chromosomes. Granted, it's the biggest one. So the stain picks up the DNA easiest. It's the most negatively charged thing there. Now, there is more going on in the nucleus. There are some charges that offset that. So overall, it's not as negative as I'm making it out to be, but it's a very negative environment. So it picks up a lot of stain. What's the second most negatively charged molecule in any cell in your body? And it should be a logical step from where we just were. If DNA is the most negatively charged molecule, you say RNA would be the next most negatively charged. It's made up of nucleotides also, and each nucleotide has a -1 charge. RNA is just a very small molecule relative to DNA. You can have very short RNA sequences that might only be a few nucleotides long, but some of them are going to be hundreds or thousands of nucleotides long. Not millions of nucleotides long, but quite a few. So it's going to be sort of the next most negative charged molecule. Now, some proteins can be very negatively charged too, but they don't usually get as negative as RNA. Where do you find most of the RNA in your cell?
Nucleolus?
Why? It's actually not true, but it's a good answer. Why is that? Why is that your answer?
I thought that's where it was stored or
It's not where it's stored,
made.
where what's made. It's a good answer.
RNA?
Steve. Not just RNA, but what's made in the nucleolus? Has to do with RNA. Ribosomes, right.
Oh, that's it.
So they're made in the nucleolus. They actually move out into the cytoplasm. So one way of answering the question, where do you find most of the RNA in your cells, would be ribosomes. But then ribosomes primarily are in the cytoplasm. So that's where you find them. So when we're staining a cell, what we've seen, the nucleus is going to stain very darkly because of all the DNA. And then we're going to see the cytoplasm picking up some of that stain also because of the RNA in the form of ribosomes there. The darker the cytoplasm is, the more ribosomes it has. What does that tell us about that cell? What does more ribosomes mean a cell does? What do ribosomes do? Yeah, they're the site of protein synthesis. So when you have dark staining cytoplasm, you can assume that means that that cell makes lots of proteins. Okay. So we're looking at two pieces of tissue here. Okay. We're looking at the pituitary gland. There's a big crack down the middle of this tissue. That's an artifact of preparing this microscope slide. You have to dehydrate tissue when you put on a microscope slide, so dehydrated tissue shrinks. And when there's a natural divide between two pieces of tissue, they'll usually pull away from each other, so that crack's there. We would not see a physical gap between the anterior and posterior pituitaries in a living system, but dehydrated tissue on the slide, you see it there. So there's a very obvious division between, on one side of that crack is anterior, and on the other side is posterior. And you can tell that one half of this is light stain and one half of it is a darker stain. One has darker staining cells, one has lighter staining cells, or lighter staining tissue. What we're interested in, though, is which of the halves are the adeno hypothesis and which half is the neuro hypothesis. So what does it mean? No, what does the root adeno mean?
Does it mean N? No.
That would be endo.
That's
What are you talking about? What is adeno? With the brain? Brain. Thank you. I don't know where I got brain. Brain is the exact wrong answer. So yes, adeno means gland. Glandular tissue is sometimes referred to as adenoid tissue. What does neuro mean? Nervous tissue. Okay. And in my head, I think that's where I was going with that. The other word here is referring to nervous tissue. So the difference between the anterior and posterior pituitary is one side is glandular tissue, the other side is nervous tissue. And so looking at this under the microscope, here we go. The two tissues stain differently. We can see that there is a difference in the tissue. So from looking at the tissue, we should be able to figure out which one's glandular and which one's nervous tissue. Now, to understand that a little bit better, let me explain what the pituitary is about. Okay, and also I want to, oops, wrong direction. go to this picture. So this is the same slide, just at higher magnification. So now we can start to see little dots that are the nuclei of these cells here and there. And in some places, so like you can see some dark spots in here, but then there's still some dark stain around those dark spots and then some lighter spaces. Over here we have dark nuclei, and pretty much everything is kind of a lighter surround. So the cytoplasm of these cells is staining darker than the cytoplasm of these cells over here. Okay. Actually, what it is there's more cytoplasm in these cells than these, but nonetheless, dark cytoplasm in these cells, not in these cells. Liz, you had a question?
Yeah, what are the big black spots?
Like these things here?
Yeah.
Dirt.
Okay.
Just dirt on the slide. There's a little bit of a fuzzy halo around them because they're actually not in focus. They're on top of the glass, and I'm focusing on this tissue that's under the glass. So let's see. I don't think the next slide is the 40X. So here's a 40X image of nucleus staining, and then its surrounding cytoplasm is darker, and there is some light space in between some of these clusters of cells, but this is really darkly staining cytoplasm. Now, if you were paying attention, you know which half of the hypothesis I'm showing you. Which is it? Well, yeah, it's the darker side, but what's it called? How do you know that? How about it says it right there?
That's why.
Right. I labeled this slide adeno hypothesis because that's all we're looking at. But the darker staining half of the hypothesis is the adeno hypothesis, which means that the lighter staining half is a neuro hypothesis. I could have walked us up to that, explaining this and gotten you to say that that was the case. And it would have been kind of a 50-50 guess for most part for a lot of given chart. I instead chose just open up this picture, and you can see the title there. It tells you it's the adeno hypothesis. And I was in fact looking, Alicia, for you to say, I know that because it says it right there.
Oh, I was trying to look for a more answer.
Make use of that information if it's available to you. If that tells you the answer, you can say it says right there. That's a perfectly valid response. What?
You don't usually tell a teacher.
I'm asking you to pay attention to that stuff and let me know if that's where you're getting the information, because sometimes that's the important clue. Pay attention to what's around you. Now, from what I've described, what does this tell you about the adeno hypothesis? It's glandular tissue. What does that mean that these cells are going to be doing?
Making proteins.
Making lots of proteins. And the pituitary hormones that are coming from this tissue are protein hormones. Okay, this is a great example of your basic endocrine gland type tissue, because most of the tissues are making proteins that are the hormones. Okay. The types of hormones that are coming from here are growth hormone. There's something called ACTH. There's something called, what's that called? I can never keep it straight. It's thyroid stimulating hormone or something like that, I think it is, which is TSH. I think that's the one that's coming from here. The book will have it correctly. I might get that confused somewhere else. But there's the gonadotropin hormones, which stimulate the ovaries to produce egg cells, or the testes to produce sperm cells, that sort of thing. Those are all protein-based hormones. Okay. Proteins, by definition, are water-soluble hormones. So all of the target tissues that these are going to have those G protein couple of receptors that I described earlier, specific for those hormones. Now, let me get out of this, and here's the 40X objective view of the other half, the neuro hypothesis. And here we can see very similar looking nuclei, but they're not surrounded by the same kind of dark cytoplasm. Now, if there is any real cytoplasm around these cells, it's very light, which means that these cells aren't making a lot of proteins. More likely, these cells really don't have a lot of cytoplasm. Now, the fact that we're in the neurohypophysis, this tells us that we're looking at nervous tissue. Now, neurons have a lot of cytoplasm, and it usually stays quite dark, which we don't see here. So we're not looking at neuron-rich nervous tissue here. The cells we're looking at here are most likely glial cells. What is present here and is not really obvious, although there's a hint of a suggestion in the texture of the tissue right here. What is present here is a lot of axons. Okay? These axons are coming from the hypothalamus. The hypothalamus makes its own hormones. Two of them, one called oxytocin and the other one called antidiuretic hormone, are released into the bloodstream in the pituitary, the neurohypophysis, the posterior pituitary. Okay. And I think this here is supposed to be a blood vessel, but I'm not positive. It's a weird looking thing. So there are blood vessels going through this tissue, and the hormones get into the blood vessels directly there. Okay. The hypothalamus, the hypothalamus makes a number of other hormones besides oxytocin and antidiuretic hormone, but those are not released through the posterior pituitary. They're released into the bloodstream, and they go directly to the anterior pituitary, where they tell the anterior pituitary to do its job. But as far as looking at the tissue, the lighter staining side over here, this is nervous tissue. This is a neurohypophysis. It's really just an extension of the hypothalamus, that the hypothalamus releases two of its hormones into the bloodstream here. The darker side over here is the anterior pituitary, which is glandular tissue. It makes its own hormones and releases it directly into the bloodstream there. Okay. It gets signals from other parts of the hypothalamus telling it what to do, but it's sort of that thing. Now, there is this little strip right here of tissue that's between the anterior and posterior pituitary. I want to point it out because it's there and you might notice it, but we're going to ignore it. It makes a hormone that that people don't really understand very well, and it's not very big. So we're not going to talk about it, but if you're looking at this tissue and you're like, well, wait a second. Okay, yeah, it's glandular tissue, that's nervous tissue. That's not nervous tissue. What is that stuff there? Yeah, it's a special little part of a gland, but we're not going to pay attention to this class because it's not very well understood. So we'll just skip over that. But that's the pituitary gland. Anterior part is glandular tissue, releases its own hormones. Posterior part is actually just more nervous tissue that releases a couple of hormones from the hypothalamus. Now I want to show you the thyroid and parathyroid glands. Now in this micrograph, we actually see three different tissues here. This tissue off to the side, this is actually muscle tissue. Okay? We're slicing through structures that are in the neck, and so we're getting a little bit of a muscle tissue that's right next to it. The thyroid and parathyroid glands are these two tissues here. And again, it's a difference between the dark staining chunk of tissue right here and the lighter appearing tissue here. Okay. And by thinking about the difference of these two tissues, we can understand a little bit about how these two glands work. So here's just the higher power picture of the two side by side. Again, ignore this stuff. It's muscle tissue. We're just paying attention to the thyroid and parathyroid glands here. Now, the parathyroid glands are these small glands attached directly onto the thyroid gland. And just from that description, you should understand that this darker little cluster here, this is a parathyroid gland. And then the bigger thing out here is the thyroid gland. Now, the tissue that makes up the thyroid gland should remind you of another type of tissue. And let me go to the lower power again. It might be a little bit more obvious. Think back to A&P1 when you're learning about tissues originally. What type of tissue does the thyroid gland there remind you of? Adipose tissue. What is adipose tissue? Fat tissue. Why does that remind you of that?
It's bubble.
This is bubble. What are those bubbles? So if it's adipose tissue, that's the fat. What is fat in a more general sense? How do we classify that? It's a lipid, right? So this isn't necessarily fat, but it is lipid. Lipids don't pick up stain. When I was describing stains, I was talking about negative molecules and that sort of thing. Lipids do not have a charge. They're completely neutral molecules, so they don't interact with stain. So they don't pick it up. Now, if we go to the 10x magnification, it looks less like adipose tissue here, because in adipose tissue, where you see a big lipid deposit, fat vacuole, that's part of a cell. And so it's going to have just a single nucleus associated with it. And you don't necessarily always see the nucleus. But here, we've got a bunch of little dots all over the place. There's a lot of nuclei here. Because the lipids here are not inside a cell. They're inside what's called a follicle, which is a little lipid environment that's surrounded by a layer of cells. Okay. And so if we look at one of these, let me get to the 40x objective where it's easier to see. So here's a big lipid deposit, and it's surrounded by what seems to be a layer of cells. Now, if you're still kind of thinking about what you learned about tissue in A&P1, you'll say, hey, that looks like a single layer epithelium. Okay. It's not. Some sources online will try to tell you that, but they're wrong. Okay. An epithelium is a lining or a covering tissue. Okay. If it's covering, it's either a simple squamous epithelium covering the outside of an organ, or it's a stratified squamous epithelium covering the outside of your body. So unless it's one of those, it's a lining tissue. Okay. And lining tissues can be simple squamous, simple cuboidal, simple columnar, stratified squamous, stratified cuboidal, stratified columnar, pseudostratified, or transitional, okay, depending on exactly where we are. Okay. Which one does this look most like? And that's what those sources are going to try to tell you. They are wrong. If you see a simple cuboidal epithelium, it's the wall of a tube, okay, like a duct. If you cut a duct, a tube, in cross-section, it'll look kind of like that. But that's not what we're looking at here. What we're looking at here is a follicle, which is a spherical shape, and it has a layer of cells coating the outside of it. And so any way we cut through that follicle, any direction the knife goes, it's going to look like that. A tube will only look like that if we cut it in cross-section, because it's a long thing with open ends on either side. So sure, these look like tubes in cross-section, but actually they're follicles, globular structures that are cut sort of along the middle. So this is not a lining epithelium. This is not appropriately called a simple cuboidal epithelium. It looks like that, but it's not. Now, glandular tissue is epithelial tissue, but it does not fit into the layer or cell-shaped structures that we talked about. So I just ran through the 8 types of epithelium. I'm not going to do that again. There's a 9th type of epithelium, which is glandular tissue. It's not in simple or stratified layers. It doesn't have squamous or cuboidal or columnar cells necessarily. We don't describe it that way. It's just glandular tissue. And we looked at glandular tissue a second ago with the adenohypophysis or anterior pituitary. Something like that we can see here or down here. And if I go to the lower power, it's a little bit easier. You can see cells kind of clustered over here, down here. These little regions of glandular tissue, or the whole parathyroid gland is like that. Where was I going with that? Oh, so that's the type of epithelial tissue. But we don't classify it based on layers or cell type. Any source is trying to tell you that the thyroid gland has a simple cuboidal epithelium in it is wrong. Just ignore that. It's not an appropriate way to talk about glandular tissue. It just appears that way because that glandular tissue is covering these follicles. Now, the reason why these lipid-rich follicles are present in the thyroid gland is because thyroid hormones, well, what we refer to as the thyroid hormones, which is only 2 of the 3 hormones that the thyroid gland secretes. They are referred to by abbreviation as T3 and T4. And that's a perfectly good way to refer to it. There's not a T1 or T2. It's T3 or T4. T3, its full name, which you do not have to commit to memory, is triiodothyronine. The tri at the beginning is where the 3 in T3 comes from. Iodo in the middle of that word is referring to iodine. And so what that molecule is it's this hormone that has 3 iodine atoms incorporated into it. That's where the triiodothyronine comes from. T4 also has iodine in it too. I don't know that it's 4 iodine molecules. It's called T4 because that's the next number after 3. The thing about T3 and T4 is that they're lipid-soluble hormones. Okay, so earlier I was talking about lipid versus water-soluble hormones. These are examples of lipid-soluble hormones. The thyroid gland has these big lipid-rich follicles because the chemistry to make lipid-soluble hormones, in this case, requires a lipid environment. So that's where those hormones are made. The cells that surround the follicle, what are called follicular cells, make T3 and T4. But there are cells that are not part of follicles. We call those parafollicular cells because they're around the follicles in the thyroid gland. Parafollicular cells of the thyroid gland make a third type of molecule, a third type of hormone called calcitonin. It is a protein hormone. It's water soluble. From the name calcitonin, tell me what it does. Yeah, it's important for calcium homeostasis. Okay. Now, calcium homeostasis is a good example of your basic endocrine what's what I'm looking for? Homeostasis mechanism. Okay. Where do we store calcium in our body? Bones. Right. When you have lots of calcium in your blood, you want to put that calcium in your bone to store it. So calcitonin is secreted from the thyroid gland when blood calcium levels are high, and it tells the bone cells to increase the storage of calcium. The other side of that, when blood calcium levels are low, you want the bone to release calcium. And that's what the parathyroid gland is for. Parathyroid gland makes one hormone, having the very boring name of parathyroid hormone, or PTH. When blood calcium levels are low, the parathyroid gland releases PTH, which tells the bones to release more calcium. So it's simply a balance there. You store calcium in your bones. When you have extra calcium in your blood, because you just ate a whole bottle of Tums or something like that, then calcitonin from the thyroid gland tells the bones to store that calcium. When your blood calcium level is low, then the parathyroid gland uses PTH to tell the bones to release that calcium. Just simply balancing that. What type of cell breaks down bone matrix to release calcium? what do we call that cell? Osteoclasts, that's right. So those are the targets primarily of PTH. So PTH tells the osteoclasts to be more active and release that calcium back into the blood. I haven't found a definitive source on what cell is the target of calcitonin, so we'll just say in the bone somewhere it has its target. There are other targets also. The kidneys also are affected by these hormones because the kidneys can help either keep calcium in the blood or pass it through the urine. Okay. And the small intestine is a target also because it can increase the amount of calcium that's absorbed from food. But primarily it's about balancing the bone's effects there. Okay. So the follicles in the thyroid gland are about making the lipid-soluble hormones T3 and T4, which have to do with other stuff. But then the parafollicular cells of the thyroid gland, the spaces in between the follicles, make calcitonin, and the parathyroid gland makes PTH. Those two hormones help balance calcium levels in the blood, working with the bone primarily. If we were to look at, well, actually I do have a parathyroid 40X objective. Okay, so there's parathyroid up close, just straight up glandular tissue. They're very small, so it's not really arranged in the clusters like we saw in the pituitary gland because the hormone doesn't have to diffuse very far to get to blood vessels passing through the tissue. In the pituitary gland, there are clusters that helps with the diffusion because there's spaces in between the clusters. But then also, the pituitary gland makes multiple hormones. So there's the different clusters make different hormones. Only one hormone is made here, parathyroid hormone, PTH. And so all of those cells are making the same thing. They're all just a homogeneous group there. Now, if I could take a little snapshot of a region of parafollicular cells in the thyroid gland and put it next to an equal size of parathyroid gland tissue, they'd be indistinguishable. Okay. They're pretty much exactly the same. What makes the thyroid gland look lighter overall is all of the lipid-containing follicles. But the glandular tissue of the parafollicular cells in the thyroid gland and the glandular tissue of the parathyroid gland are pretty much identical in appearance. Okay. Someday I'll have to make a little side-by-side comparison of this, but just take my word for it instead. Now, very important that you were paying attention to how I said that. Parafollicular cells of the thyroid gland. Parathyroid gland. They both have para in them. But the parathyroid gland is around the thyroid gland. Parafollicular cells of the thyroid gland are around the follicles of the thyroid gland. So parafollicular. They're around the follicles. So in the thyroid gland, you have follicular cells and you have parafollicular cells. Follicular cells make T3 and T4 using the lipid environment in the follicle. Parafollicular cells make calcitonin, which is part of calcium homeostasis, like I just mentioned. So when you see para in a word, don't just assume that it means parathyroid gland, because it might be parafollicular cells of the thyroid gland. Okay. Now I want to move on to looking at the pancreas. So before we were looking at two different tissues of the adeno and neuro hypophyses, or two different tissues of the thyroid or parathyroid glands. Now we're looking at two different tissues within one gland, in the pancreas. And the distinction here is between dark and light staining cells, or tissues, is endocrine versus exocrine function. Endocrine means discrete within. Hormones in the bloodstream. Exocrine means to secrete outside. Now, a sweat gland, like we we're talking about in the first half, is a very obvious example of an exocrine gland. You sweat to the outside surface of your skin. But digestive glands or respiratory glands or reproductive glands are also exocrine. The space inside your digestive tract or inside your reproductive tract or inside your respiratory tract or inside your urinary tract for that matter, those are all considered outside your body in an immunological sense. A pathogen will try to get through your skin or through your digestive mucosa. And I'm not going to point so directly at my nasal mucosa, but those barriers are between the inside of your body and the outside of your body. So when we talk about secretions, exocrine secretions aren't just outside your body in your own sort of sense of self outside, but also outside of your body in the immunological sense across the mucous lining of your digestive or respiratory or reproductive or urinary tracts. So here we're looking at the pancreas. The exocrine parts of the pancreas are the ones that are making digestive juices that get secreted into the digestive tract, specifically into the lumen of the small intestine. The endocrine parts are the ones that are making hormones that go into the bloodstream. And the exocrine tissue far outnumbers, that's not the right word, but there's a lot more exocrine tissue than endocrine tissue. There's a lot more dark staining stuff than light staining stuff. So right here, for instance, there's this little island of lighter staining tissue floating in the bigger, darker staining region right there. Okay. If we look at that at a higher power, okay, here's that little light stained area here, and there's darker clusters of cells around it. Okay. It's a little more detail. We can start to see nuclei here, but still we can see a lighter area versus a darker area. And this is the 10x objective. And if we move up to the 40x objective, all three of these I've been pointing out the exact same piece of tissue. So here we see it at an even higher power. This little lighter staining island of tissue in the midst of this darker staining surrounding region. Now I'm using the word island on purpose. This is what's called a pancreatic islet. An islet means a little island. So it's floating in the sea of the bigger pancreas. That's where the name comes from. The lighter staining tissue is the endocrine tissue, and the darker staining stuff surrounding is the exocrine tissue. Now, the reason why the pancreas gets a lot of attention as an endocrine gland, even though that's a very minor part of what it really does, is because the hormones that are released here are pretty well known, or at least one of them is. It releases insulin, which I'm sure you've all heard of before. It also releases glucagon, which is the antagonist, if you will, to insulin. Okay. Now, I'm using antagonist in a literary sense. It's the bad guy. It's the other side of the coin. When we're talking about signaling molecules and receptors and something like that, antagonist has another meaning, and that's not the meaning I'm using here. So from cells in the little islet here, we have insulin and glucagon being released. And they're responsible for regulating blood glucose levels. So a second ago, I was talking about calcitonin and PTH regulating blood calcium levels. Insulin and glucagon regulate blood glucose levels. And it works kind of the same way. Okay. When blood glucose levels are high, a certain type of cell here releases insulin, and that lowers blood glucose levels. When blood glucose levels are low, another type of cell here releases glucagon, and that raises blood glucose levels. Now, this is a great place to get the idea of target cells across. Insulin to lower blood glucose levels is going to tell every single cell in your body that there's glucose in your bloodstream and it needs to take it up. So every single cell in your body has insulin receptors on it. Everything in your body is an insulin target because they all need glucose. Glucagon, however, only targets the liver and a couple of other minor tissues, but we're going to concentrate on the liver. So the liver has glucagon receptors on it, but nothing else does. Oversimplified. That's essentially the case. So if you have any old cell, you can put insulin on it and it'll react. But unless it's a cell from the liver, if you put glucagon on it, nothing's going to happen. It's not a target for glucagon unless it has the glucagon receptor.
Is insulin and glucagon, they're two different forms of medication?
Sorry?
They're like two different forms of the medication?
No, they're two different molecules. They're physiologically working opposite to each other. When blood sugar levels are high, insulin causes any cell in your body to take up glucose. When blood glucose levels are low, glucagon tells the liver to release more glucose. At the molecular level, they're not at all related. It's just at the physiological level. Now, the way that glucagon works, what the liver does in response to glucagon, is it breaks down the starch-like storage molecule called glycogen, which is just a big network of glucose molecules, breaks that down, releases the glucose into the blood, and the glucose levels go back up. And it tells the liver to make new glucose. Now, glucose is made through photosynthesis, which we don't do. Our liver does not photosynthesize to make new glucose. What the liver actually does is it reconstitutes glucose from the waste products of when glucose was broken down previously. Primarily, that's lactic acid. So lactic acid has 3 carbons in it. Take 2 lactic acid molecules and you put them back together, you get the 6-carbon molecule of glucose. Sort of. There's more to it, but that's basically what's happening. It's making new glucose out of the leftovers from when it's been used before. It's not a very effective way or efficient way to get blood glucose levels up, but when it's necessary, it's there to be done. Glucose. Oh, no, insulin, that's where I was going with this. Insulin does a little bit more than just telling cells to take up glucose. When do you have high blood glucose levels usually? When do you have high blood glucose levels usually?
You need a lot of sugar?
When you eat. When that
Yeah, you don't have to.
Just,
Yeah.
there's carbohydrates in most everything we eat.
Sorry, normal pancreas would make that go back to normal and it's already a problem with it. It would-
Right, so normal function, you eat food, blood glucose levels go up, cells in the islet here release insulin, tells all your cells take that glucose out, goes down. Diabetes is when that doesn't work. Insulin's not doing its job. A few reasons why that can happen, but we're not going into that. If your blood glucose levels are up because you just ate, then your blood lipid levels and your blood amino acid levels are also going to be higher. Those are other nutrients that are going to be interfered with. So while glucose is really responding to the blood glucose, insulin is really responding to blood glucose levels, it's also going to tell cells to take up the amino acids and lipids that are present. Just an efficient use of that signal. If you've eaten, then you probably have all of those nutrient molecules in your blood. So that one signal really signals all of those to be taken up. Insulin will also increase protein synthesis to make use of the amino acids, and that sort of thing. So there's a few things that insulin does beyond just regulating blood glucose levels, but it's because all of that stuff's going to follow when you eat and you have that stuff on hand. So it's going to do that. Now, when I was originally talking about this, I just said one cell here will release glucose and another cell is going to release glucagon. Those cells have names. There are alpha cells and beta cells. Now, alpha cells release glucagon. Beta cells release insulin. The way that this tissue is stained, we can't tell the difference between alpha cells and beta cells. We could use a different type of stain that would highlight the differences between them, but we can't be sure here. However, there is a rule of thumb that we can use. Alpha cells tend to be on the outside of the islet, and beta cells tend to be in the middle of the islet. So if I'm pointing to a cell right here, it's most likely going to be which of those two types of cells? Beta cell, because it's in the middle of the islet. And if I was pointing to a cell out here, that would most likely be which? Alpha. Which would release which hormone? Glucagon. So while we can't actually tell the difference between the cells with the type of stain we have here, that rule of thumb is a pretty good guide to what's going on. So that's the pancreas. Oh, actually, sorry. Let me go back to talk about the exocrine stuff a little bit. In the digestive system, we'll talk about the pancreas in more detail. But that darker staining tissue that's surrounding the islet, that's exocrine tissue. And what it does is it makes enzymes to help break down the food that we've just eaten. There is some obvious relationship between the endocrine and exocrine functions in the pancreas. They're both dealing with what happens when we eat. So we need to break down the food that we've taken in, and we need to take up those nutrients into the cells that we have in our body. So they're really kind of tied together. The reason why the exocrine cells are darker is because they're making a lot more protein than the islet cells. Insulin and glucagon are both proteins, but we only need enough of that signal to tell our body what to do, which is pretty small. But the exocrine cells are really dark because they're making lots and lots of enzymes, which are also proteins. We need a lot of enzymes to break down all of the molecules in the food we eat. So the really dark cells are really busy making lots of enzymes to be secreted into the digestive tract, whereas the lighter endocrine cells here are just making enough insulin or glucagon to get the signal out so that your body will respond to it. If we were to compare the exocrine tissue here with some of the other exocrine glands that we have, it would probably look really similar, but next to the very dark staining exocrine tissue here, it looks pretty light. Okay. So that's more of a trick of your eyes than anything else, but it's the same kind of glandular tissue that we've been talking about all along. Okay, I want to finish up by talking about the adrenal gland. Okay. So in the first half today, I mentioned the adrenal medulla, which is tied to the sympathetic system's activity. It gets input from a preganglionic sympathetic fiber, and it releases epinephrine into the bloodstream. And epinephrine acts just like norepinephrine, and it causes the same changes as any postganglionic sympathetic fiber would. For the endocrine system, the adrenal gland, the part of the adrenal gland we're interested in is called the cortex. Medulla basically means middle, and you can kind of see where the two words are similar. Cortex means the outside of something. And as far as English is concerned, the word that's a cognate for cortex is cork. Okay. Cork is the bark of a tree. So when we're talking about the cortex of something, we're talking about the outer covering of something. That can be a little misleading in this picture because the cortex is all of this part of the gland. Okay. So the light sort of oval center here, that's the adrenal medulla, the adrenal medulla. And then as soon as we see a change in the staining quality, we go from this lighter tissue to this darker tissue here. From that point out, the rest of it is the adrenal medulla. The adrenal medulla, sorry, I said that wrong. The adrenal cortex is making steroid hormones, which is another type of lipid-soluble hormone. However, unlike the thyroid gland, we don't have follicles here. Steroid hormones are made from cholesterol. Right? So we don't need reserves of lipids for the metabolism of lipid hormones here, like in the thyroid gland. Cholesterol actually is found in most cell membranes. So these cells are going to have a bunch of cholesterol available to them in their cell membranes. Just pull it out, modify it, and secrete it. We can see, however, some difference in staining across the width of the cortex. Let me go to the next picture. Whoops, sorry, wrong direction. Yeah. So here's the outer surface, and we can move all the way down to right here. That's the edge of the medulla cortex border. And as we look across the tissue, we have very different staining qualities for these cells as we move across. And we see different layers of tissue here. Now, the difference that we can see here anatomically, the staining differences, corresponds to functional differences. The cells in these three different layers we see here secrete different hormones. From the outside in, these layers are referred to as the glomerulosa, and I'm going to write all this down in a second. The zona glomerulosa, the zona, fasciculata, and the zona reticularis. I'm suddenly drawing a blank, and let me just verify that. I got it right, yay, okay. So here it is laid out in this picture. Let me just write it down here. Sorry, I want to skip a little space between these because I need to add to it. Zona. What was that? Shoot. Reticularis. Oh, great. I messed it up this time. So those are the three layers. Now I've left some space in there because I want to tell you what's secreted from each one. Okay. The zona glomerulosa is responsible for making what are called mineralocorticoids. Corticoid means steroids made by the adrenal cortex. Okay? And we're going to see that repeated in these other two layers. Minerala is referring to that these are hormones that are responsible for regulating mineral levels. Okay? Sodium, potassium, well, we've already talked about calcium elsewhere, but those sorts of things. That's what it's responsible for doing. Okay? The zona fasciculata makes what are called glucocorticoids. So again, it ends in corticoid because it's a steroid made by the adrenal cortex. What does gluco refer to? Sugar. So glucocorticoids are responsible for regulating some aspects of glucose metabolism. Now, we just talked about insulin and glucagon, which have to do with glucose levels in the blood. But these are going to be hormones that regulate what cells do with glucose, and to some degree, what the liver does with it too. And then the zona reticularis is responsible for gonatocorticoids. Now, most places will not use that term to refer to these. More often, they'll just be called sex hormones. Okay, so that would be androgens like testosterone or estrogens. Okay, now estrogen is not a hormone itself. Estrogen is a class of hormone. There are three main estrogens: estradiol, estriol, and estron. So they all have estro in them, but as a group, they are estrogens. Androgens are the antithesis to that. Okay, and testosterone is a type of androgen hormone. Okay, why testosterone gets mentioned on a regular basis as the specific hormone name, but then the estrogens are usually just lumped together, and to the point where people usually think that estrogen is the name of a hormone. Estrogen is the name of a group of hormones, but gonadotropins would include all of those. Okay, androgens and estrogens. So again, corticoid, steroids are made by the cortex, and gonado is referring to the gonads or the reproductive glands. So the sex hormone is going to have a role to play there. Now, there's a little quirk about the zona reticularis, which it also makes glucocorticoids. And that little quirk actually shows up in the anatomy. So let me go back to this picture here. Oops, picture here. There's a very distinct line separating the zona glomerulosa from the zona fascicularis. But the line between the fasciculata and the reticularis is a little vague. Okay, sort of blends together. We can't draw a really obvious line between them like we can here. And that is really just saying that all of the cells in both these layers make glucocorticoids, but the darker cells also make gonadotropins. So there's sort of an additional function on top of that. Not a very distinct transition like we see here, or if I move the picture over a little bit, so here's the fasciculata, the reticularis, and the border between the medulla and the cortex right there. So there's a very distinct line between where the corticoids are made versus epinephrine in the medulla. But or there's a nice distinct line between where the mineralocorticoids and the glucocorticoids are made. But there's not a very distinct line here because the function kind of blends from one layer to the next. Okay. And so when you can see an anatomical difference in tissue like that, it sometimes means that there's an underlying functional point to it also. So again, given the time available, I would just wanted to concentrate on using these micrographs to talk about some specific glands and some of how it works. I didn't go into everything about the book. The book does talk a bit more about a few other examples of hormone regulation, so please pay attention to those. It goes into more detail about the insulin stuff, and I think the book goes into the calcium stuff I talked about too. But then it mentions a few other things in the more detail here. And also the relationship of the hypothalamus and the pituitary. So please pay attention to that stuff. We just didn't have the time to go into it as much depth today. I will double check the due dates, but keep in mind that all of the autonomic system and endocrine system material, including the lab quizzes in both those unit folders, are due before next week. So, otherwise, have a nice weekend. No more pre-unit assignments.